on and talk about heterochromatin condensation. And heterochromatin are just these very dense regions of the DNA. And so first I'll give you a bit of the background on the biology of this system. I'll talk to you about our model for heterochromatin, and then I'll talk to you about some results that we have. So as I mentioned at the start, DNA is packed across a wide variety of length scales. And the DNA here is going to be wrapped around these proteins called histones, and then it can be packed into these higher order structures like these called chromatin fibers. And this is sort of the area that I'm going to be focusing on here. If you look at a picture of the nucleus, and you'll notice that there's these very dark regions in this image, and these are very dense regions of the nucleus. And these regions are called heterochromatin, and they often code for genes that are turned off or repressed. It's basically things that the cell doesn't need to access. And so the question I'm going to be addressing is how are these distinct DNA heterochromatin regions formed, and then how is that information passed on to future generations of the cell? Now there's a number of chemical tags that can decorate the proteins that the DNA wraps around. These proteins called histones, they can have acetylation marks, they can have methylation marks. And these marks may code for different degrees of condensation within the nucleus. Now these marks are called epigenetic, and that's because they're associated with the DNA region and they can be inherited along with that DNA region. And so these marks can then be, in the future daughter cells, can still be associated with these regions and pass that information along. Now there's a particular mark I'm going to be focusing on today, and that's the methylation of the histone 3 tail at lysine 9. And that's shown in red here in this crystal structure. And the nucleosome refers to the whole histone DNA complex, shown here. Now, throughout the talk, I'm going to be using this representation for the nucleosome. And essentially, these elbows here represent the tails, and they can be up to trimethylated. And so that means they can have be either unmethylated or have 3 levels of methylation. Now there's a particularly important protein, and that protein is called heterochromatin protein 1, which binds these methylation marks in these regions in the cell. And it's known that it binds most strongly when it's trimethylated. And those are the marks found commonly within these heterochromatin regions. Now this protein, disruption to this protein is found to disrupt the formation of heterochromatin, as well as targeting this protein can actually induce open regions to then condense. So it's thought that then HP1 might be playing a role of sort of helping hold these regions together. And it's been suggested in the literature because HP1 can interact with itself, it might create these sort of bridges that help hold different chromatin regions together. Now, this is not a static picture, because it's been measured that HP1 can actually bind and unbind very rapidly, even in heterochromatin regions. And so, this isn't a static structure, but we know that the binding of HP1 is at equilibrium throughout the whole nucleus, even in the heterochromatin regions. And so this suggests that any preference for HP1 going into heterochromatin regions, or these highly methylated regions, has to be driven by thermodynamics. And so now I'm going to talk to you about our model for HP1 binding. And we're going to start with low energy chromatin fiber structures. Now, as I mentioned, it's not known the exact structure within the nucleus, but we can look at a range of different structures. And all this is based on work from someone else in my lab, Elena Koslover. And she looked at the low energy structures based on the DNA linker behavior, as well as the sterics of these nucleosomes. For today, what I'm going to be focusing really on is the location of the tails, which is where the HP1 will bind. And to simplify the picture a little, I'm going to look only at the case where they're either unmethylated or trimethylated. Now, HP1 can then bind to these tails, and you see the orange lines indicate the interactions that can occur between HP1 molecules. If we zoom in, we can now define that there's going to be some distance over which the HP1 could interact. That is, if the two tails are very far away in the structure, the HP1 couldn't interact with them, but HP1 bound that nearby tails could. So we define a distance alpha, which is the sort of maximum distance over which these two proteins could interact. And we can define a connectivity between the different tails based on how far away they are. And connected sites that are within this distance alpha, will give they'll equal 1 in this matrix, and otherwise they'll be unconnected, they'll be 0. Now ultimately, the picture we're trying to capture here of this crowded cellular nucleus, we're going to model by having fibers packed together in these sort of repeating boundary conditions where we have connections within the fibers as well as connections between fibers. And so our model is going to account for two energies for HP1. The first is the binding energy. So we have HP1 that can bind to these tails, and if the tails are methylated, it'll have a binding energy of epsilon m, and if they're unmethylated, a binding energy of epsilon u. And of course, because it favors the methylation mark, the epsilon m term is going to be larger. Now, if the HP1 is bound to two tails that are within that cutoff distance alpha, then there'll be favorable interaction energy that's given by J here. So next we want to calculate our partition function. And our partition function is essentially just a weighted sum of states. So if we have n different binding sites for our HP1, then there's 2 to the n different states of the system, because each site can either be bound or not bound by HP1. And so if we weight that by the probability, which is given by the exponential of the energy, and then we have this chemical potential term. And the chemical potential accounts for how much HP1 we have in solution. And so the more HP1, the more favorable it will be to bind. From this, we're going to calculate the free energy, and as well as the average amount of HP1 binding. Now, before we can do that, we have to consider the disorder in the methylation, where you could have the methylation marks distributed in any number of different ways within a region. So if this region is 50% methylated, you could have the methylation marks in a number of different places and still have a 50% methylated region. And so we need to average over all these possibilities. And so we can do that, and then we're going to use a technique called self-consistent field theory that I won't have time to get into today. But the basic idea is that we're going to turn the summation that you saw on the previous slide into intervals and then make systematic approximations. The lowest order approximation is called the mean field result. And basically, you average over all the interactions to create an effective field that fields. And then we can look at fluctuations in HP1 binding away from this result. And that's the results I'm going to be showing you today. So now I'm going to move into the last part. And before we can get a sense, we need to get a sense of what our energetic parameters ought to be. And so if we look in the literature, there's been experiments done from this Conzio et al. Paper, in which they looked at HP1 binding to mononucleosomes. And that's the fraction bound versus the concentration of SY6, which is just a similar protein in yeast. And so we can model the data by looking at the fraction bound as the different states that a single nucleosome can take, overall, that have HP1 bound, over all possible states that it could be in. And based on the energy weights for each of these states, we can then calculate a fit to this curve and get our parameters. And so the parameters we come out with are an interaction energy, J, which equals 4 kT, a binding energy, with the difference in the binding energy between the unmethylated and trimethylated state, which is 1.5 kT, and finally a reference potential. And this is to basically convert our chemical potential to concentrations that are measured within the cell. So now we want to go from this mononucleosome picture to looking at these packed fiber arrays, as shown here. So let's just take one representative structure shown in here, all packed together. And the only thing we don't know yet is what the value of alpha should be, that cutoff distance, which you can see here. And so it should be reasonable enough for the two HP1 molecules to span. And so we start with the alpha equals 6 nanometers case. And if you look at the binding of HP1 versus the concentration, you'll see it rises smoothly here. Now if we just look at a slightly larger value of alpha, 7 nanometers, in the purple curve, you see a much different behavior. You have this very slow increase of HP1 binding, and then a sudden discontinuous jump. And this jump here is characteristic of a phase transition. Essentially what it means is that it's unfavorable to keep staying within this unbound state, and so it's going to jump up into a highly bound state. Now alpha is only changing the amount of connections within these fibers. So if we look at this two-fiber diagram here where the squares correspond to different binding sites, you can see the lines correspond to different connections within this fiber. And we can define, some sites have more connections than other sites, but we can define an average number of connections, Z. And so alpha is just changing Z, the average number of connections within this fiber. Now at each site, there'll be some number of connections times an interaction energy per connection, J. So we define this quantity Jz, and we can see that Jz is very important in determining the phase behavior here that we see. So for our large values of Jz, there's a jump, and you have the phase transition, but when you have these smaller values, you don't see that. And just to look at an intermediate value of alpha, we see that again, for this Jz value, 8 kT is large enough to still have a phase transition. And so this total cooperativity index that Jz is really measuring is the key parameter in determining whether or not you will have a phase transition. And ultimately, having a phase transition is critical because phase transitions are what allow the cell to create these very clearly defined regions where we saw that those dark areas separated from those light areas. And that's controlled only if you have these phase transitions happening. Now we can look at the same structure as we did before, same picture, and let's just look at a different fiber structure. So in this other fiber structure here, here's what this one looks like. And it's you can see a different packing arrangement, different alignment of the nucleosomes. And what you notice is that for the same values of alpha, you have different degree of connectivity, and therefore you have the phase transitions look different. If you just look at the 16 kT case here versus this one here, what you can see is that although there's some sort of differences here based on the heterogeneity, that is, these sites might have some sites that are very connected and some that are very sparsely connected, whereas this is a much more homogeneous degree of connections. And that gives it this characteristic drop here. The actual location of the phase transition is very close to the same for the same value of Jz. And so I'm going to be focusing most of the talk now on a very homogeneous binding picture that is homogeneous connections throughout. But we can always relate back to any of these different fiber structures by relating back the total cooperativity index, Jz. So now let's look at the effect of methylation. So in a region in which you have highly methylated and highly unmethylated, 100%, you can see that at very low concentrations of HP1, we're not going to get any binding in these arrays. Now, if we increase the concentration of HP1, we're going to get selective binding only to the methylated arrays. And that's shown here at these points Y. And so this area of concentration here, this is defining a window in which HP1 will only be enhanced in the heterochromatin regions. And this is the window we'd expect the cell to want to operate in to selectively have HP1 bind in those regions. Now, increasing the concentration further to point Z, you can get binding even in these unmethylated fiber regions. So this picture here is really just parceling out the phase space. And we can look at that. There's going to be a section where we have no regions bound. There's going to be this narrow window here where only the heterochromatin regions are bound. And here you can see the points xyz from before for a given value of jz. And then there's going to be a region where all fibers will be bound. And here in yellow is just to show you where the physiological concentration range of HP1 has been measured. And so the cell in general will want to operate within this window here in order to keep only heterochromatin regions bound. But it might want to decondense these regions at different times in the cell cycle to get access to them. And it can do so by changing the HP1 concentration. And so now we can see why this binding energy in this window is so small. The cell prefers this small window because it allows it to easily change the concentration and change the condensation amount. If instead the binding energy were much wider, it would be impossible for the cell to be able to change the concentration of HP1 and decondense certain regions during different pieces of the cell cycle. So next we can ask, is there some amount of methylation that we need within a region to get these heterochromatin regions to form? And is there some cutoff amount? And so if we look at the HP1 binding versus the amount of methylation, we see in the blue curve that we have the value of Jz is very low, and so it's not as cooperative of a system, cooperative binding system, and then the blue curve just rises very gradually. But in the red curve, Jz equals 5 kt, you see the sudden jump characteristic of a phase transition. And so here you can see the value of the phase transition, because if instead we had the picture of a low cooperativity, then these regions would just blend into one another, and you wouldn't create this clearly segregated heterochromatin domains that you see in this picture. Instead, this defines an amount here, a threshold, that the cell needs to keep these regions above. If it has a methylation above this amount, then those regions will form heterochromatin, otherwise they'll be more accessible regions. So the last thing I want to talk to you about is the flexibility of these tail regions. So this is the nucleosome crystal structure picture for the locations here of the binding sites for HP1. Now, these tails are flexible, and simulation work from Gaurav Arya has looked at how these tails can flex, and I've chosen here two different representative positions for the tails. And we can see what the effect of these different tail flexing outward are. In our fiber structures here, all the tail configurations like these will have the binding sites pointed inward. So what this means is that there's a lot of connections within this fiber. But as the tail flex outward, there'll be less connections, but we'll see that this will allow for connections between fibers. And that you can see in these diagrams here. In this case, there are a lot of connections between each binding site within the single fiber, but nothing between the fibers. And it's only when the tails are allowed to flex outward from the fiber can you get these green lines here, these connections between fibers. And the key point here is that by allowing the tails to then interact with neighboring fibers, you can form these aggregated domains. Otherwise, if they were only pointed inward and couldn't interact just within the fiber, you could not form these larger structures. Okay, so now I just want to summarize the second half of this talk. I talked to you about the role, different key in vivo pieces for allowing heterochromatin regions to form. We discussed an enhancement window for HP1 binding, and how that if you have enough cooperativity in your system, you can get this methylation threshold to form to separate heterochromatin regions from other regions. And finally, how tail flexibility allows for connections between fibers and allows these aggregated regions to form. So I just want to summarize the whole talk. In the first half, I went over the key effect of protein flexibility and how this alters the energy landscape that we saw for this looping reaction. And more information can be found in these upcoming manuscripts. And then in the second half, I talked to you about what are the in vivo conditions necessary to form these heterochromatin regions, these very dense, packed regions. And ultimately, we're connecting the marks in these regions to a mechanism for condensation. Now, there's a lot of interesting directions that this work can go. And our work in the first half made very clear testable predictions that you could look at other proteins or altering the structure of lactic repressor to see how that will affect the kinetics. We could also look at simulations of this looping process. And this is work that I didn't have a chance to discuss with you today, but I've done some work on simulations to make them more efficient to capturing the looping process by changing the number of beads during the course of the simulation. So you only simulate the finest levels when you need to simulate those finest levels. We could also look at simulations for the chromatin fiber case. And this is work done by Shafan in my lab. And he's looking at aggregating these different regions of different chemical identities, but is very analogous to the chromatin fiber organization that we see. And of course, there are many other examples of chromatin packing that have they could have very analogous behavior. For example, X chromosome inactivation also involves very dense formation around the X chromosome. Okay, so with that.